Okay, again, good morning everyone. Good morning, grade 10. We are still in the middle of our discussion for our topic. Let me share my screen. Let me share my screen. Okay. Okay. Um, Regarding the prime 5 prime end and 3 prime end, this is actually remember that your sugar there has 5 um carbon molecule. It's a 5 carbon molecule, right? Grade 10.
Yes,
Okay.
yes.
Yes.
So
Yes.
your prime end is connected to the 3 prime end of the other backbone or phosphate sugars. Okay? Remember that these phosphate sugars connect by 5 and 3 in order not to be parallel. That is why it is an anti-parallel or anti-parallel with up and side direction. Let's continue. We're done with the messenger, ribosomal, and transfer. DNA and its relation to heredity and diversity. Can you see my shared screen still?
Yes.
Yes.
Okay. Humans grow and develop according to the instruction of our genes. The gene is a segment of DNA that may code for protein for hair, insulin, which controls the glucose or sugar in the blood. And the DNA molecule also is a very long and may contain hundreds, even thousands of genes. The DNA in your genes is responsible for uniqueness and shares some features of your family member. However, before the information of your genes becomes useful, the segment of DNA should be cracked, okay, or decode. The code dictates the order by which the amino acid will be assembled to form a protein. Okay? Again, remember, when you decode your DNA into RNA or messenger RNA, it will convert thymine into uracil. Okay? And connects with or pairs with adenine. Therefore, codes the structure of protein and protein structure in turn will determine the role of in your body. It must be remembered all throughout the cell or must carry the genetic information. This is why before a cell divides during mitosis, are you familiar with mitosis? Grade 10?
Yes.
Okay. What is mitosis? Anybody?
The process in which a single cell divides into two.
Okay. How about meiosis? How about meiosis?
Meiosis, sure.
Yes.
Okay. Meiosis.
Okay.
I don't.
Phases is does mitosis contain or has?
Two.
We have how many phases? This is also a lesson from grade 9.
Four.
4, yes. 4, correct. Okay, it's 4. But if you include interphase, it will become
Five.
5. Okay, some of the books, some of the authors includes the interphase. Okay, to make it 5. And During interphase, it is said that this is the point where the cell actually prepares itself for possible mitosis. Okay? Supposed to be, they coined interphase as the relaxing point, or the point wherein the cell is not moving anything. But upon observation, during when the cell is in interphase, or cell at interphase, it is actually busy. The cell is busy preparing for itself for a possible mitosis. Okay? So the hierarchy are interphase, prophase, metaphase, anaphase, and telophase. Okay? Did you discuss about this in your lower years? Or
No, we
okay.
haven't discussed that.
This is a continuation, by the way, by your cell division at the same time genetics. This is a combination of genetics, a combination also of um, cell division, and a combination of diversity, biodiversity. That is why. So let's um, take a look at the overview of mitosis. Okay, when the cell is in when the cell is in interphase, it is preparing itself for possible um, mitosis. So the chromosomes there becomes darkened and prepare itself for possible mitosis. In the prophase, the chromosome starts to move. Okay, the chromosome starts to move, and aligning also the spindle fibers and the centromeres. Remember the centromeres? Centromeres, grade 10. This is one of the organelle responsible for pulling the chromosomes apart. Grade 10.
Mm, not
Haven't
familiar with it.
haven't talked about that before, sir.
Okay. Because we cannot continue unless you understand what is with mitosis. Okay? Because mitosis is not just a um, simple event. This is a major event. Okay? This is the reason why we can actually carry those genetic informations. Okay. Just give me a minute. I will look for my slides in order for you to understand the mitotic cell division and the meiotic cell division. Let me check on my slides. Are you still with me? Let's continue.
Yes,
Yes.
sir.
Okay. Can you see my shared screen?
Yes.
This is actually my PowerPoint presentation. I was still in college. Okay. Let's tackle mitosis first in order for you to understand clearly the how these genes are actually attributed, how these genes are actually distributed all throughout the progeny. Okay. Mitosis. Eukaryotic cells typically divide by mitosis or indirect cell division. Mitosis because when you say eukaryotic cell, what is eukaryotic cell by the way? Are you familiar with eukaryotic and prokaryotic cell?
Not,
Okay.
not familiar.
When we say eukaryotic means true. True karyon, karyotic or karyon means nucleus. It has a true nucleus. True because the nucleus there is actually wrapped with a membrane bound protection called the nuclear envelope or nuclear membrane. Nuclear membrane. Okay? It actually protects and help the nucleus perform its function. Right? Let's have the functions of or the function of mitosis to construct an exact copy of each chromosomes. So meaning to say, whatever is the mother cell, it should be transferred or it should be identical to the daughter cells. That is the function of mitosis. The second one is to distribute through cell division or division of parent cell an identical set of chromosome to each of the daughter cells. Okay? If it is not identical to the parent cell, therefore, there's a problem with that cell. And it can cause harm. Like for example, if your body creates a defective cell, it can actually create something. Like for example, um, cysts. Okay. The cyst or polycystic or cysts, commonly cyst, as what we actually coin it, it is an abnormal cell. If you're going to examine it, it is not complete and it has also different set of genetic characteristic. The first phase, the one that I'm telling you, if you're going to add interphase, it's 5, but usually it is not added. But we need to consider interphase as is not actually part of mitosis, correct? But this is a phase between cell division because the cell now is preparing itself for possible mitosis. So during this rest period, we coin this rest period because the cell is not actually moving. If you're going to look at it, it is not actually moving, but it is preparing itself for possible cell division. During this rest period, normal functions and other than reproduction take place. At the interface, the chromosome appears to be jumbled mass of threads called chromatin. So it becomes now what? Blackened, darkened. So if you're going to look at it under the microscope, blackened siya, dark. Okay. Later on, don't worry, we will try to see what is a cell at interface look like under the microscope. One or more nuclei are present in the nucleus because it is preparing itself for possible mitosis. In animal cells, two pairs of centrioles are just outside the nucleus. The one that I'm telling you, it is preparing itself for what? For pulling out of the chromatids. DNA replication occurs at interphase before mitosis actually begins. So this is typically the preparation stage for possible mitosis. The synthesis of proteins also occurs in this stage. Stages of interphase that we will not discuss this one. This is higher. But this is really interesting. Let's focus on the cell division. Okay. Okay. So this is a cell at interface. Can you see it clearly?
Yes.
Okay.
Yes, sure to do.
This is a cell at interface. If you're going to look at it, the centrioles are actually moving towards the opposite direction, padung sa isig kakilid, to pull the chromatin in opposite direction. Okay? This is actually under the microscope. We actually do this one. We did this one. Hopefully also we can do it. But it's impossible for this situation, the pandemic situation. But that is actually the real, the actual specimen under the microscope. Okay? Mitosis could be divided into two phases. We have karyokinesis or nuclear division. Karyo means nucleus. And from prophase to telophase. And cytokinesis or cytoplasmic division. Okay? Okay. This is now but that is not the correct spelling, ha. It's the wrong spelling. Usually, they misspelled this one before. This is an old copy actually. We get this one from the old um, sources, but this is much clearer. You can actually see how these chromatids are actually moving. Now, if you're going to look at prophase, it is scattered and the nuclear envelope actually now broken. In the metaphase, as you can see, the chromatids are actually in line in the equatorial line of the cell. In the anaphase, the chromatids are actually separated to each other. And the telophase, which divides now the cell. So it now creates an envelope or a cleavage. We call that one cleavage. This one. This one. This is actually the cleavage. Okay. This is one of the illustration also on how cell division works. Mitotic phases or karyokinesis and cytokinesis could be broken into mitotic phases. We have prophase. The onset of chromatids begin to shorten until thicken becomes clear and distinct chromosomes. Prophase. At later part will be just doubled and joined by a centromere. Prophase. Let's move now to metaphase. Okay. The second part is metaphase. Okay. Chromosomes attached to spindle fibers have moved into somewhat a midline position or in the equatorial line. Okay. This cell equator is sometimes designated as the metaphase plate. Okay, this is now the cell at metaphase. If you're going to look at it, it is actually aligned at the center of the cell. Anaphase. The beginning of anaphase is marked by the separation of chromosomes in one member of the pair of chromosome moving towards the one end of the pole and the other is moving in the opposite direction. So, nagpalayo. They are already separated from pole to pole. This one. This is anaphase already. That's anaphase. Okay? Early because as you can see, it is not completely separated. Telophase. This is marked by the arrival of the chromosomes at the poles. So, it is actually distinct to lose their they begin to lengthen and resume their interphase condition. Okay? And it has also cleavage. Okay? It has already cleavage. Are you still with me? Great.
Yes.
Yes.
Yes, sir.
Okay? The end of telophase is the start of cytokinesis. Okay? Meaning it creates now the cell itself. So that is how cell division works. Was that really long? It is still in the mitosis. Let's continue. Let me share my screen again. Okay. This is why before the cell divides during mitosis, the chromosomes are doubled so that the daughter cell will have the same copy of the genetic material. That's the purpose. Replication is the process by which a copy of original genetic information is duplicated so each new cell can receive exactly the same information as the parent cell. The sequence in one strand of DNA will automatically serve as the template to supply the information needed to produce its partner or complementary strand or complementary pairs. The template and the complementary strand will now make up a new double-stranded DNA identical to its original strand. By the way, do you have any questions so far?
None so far.
None so
None.
far.
None.
None yet.
Okay. Please let me know if you have questions so that we can I can assist you with that, with your questions. For replication to occur, the twisted DNA must unwind and separate. This is made possible by breaking the hydrogen bond to the paired nitrogen bond with the aid of enzymes. Free nucleotides in the cell find their complementary bases pairs or base pairs, and enzymes will again bind these nucleotides to make a complementary strand. Okay, let's stop with copying the recipe, or let's continue with the copying recipe by next meeting. We done a lot today, and hopefully you get something out of our simple discussion. Okay, science is not that broad, it is not that complicated as long as you understand the concepts. The one that I discussed a while ago is actually a topic of the previous years. But in our department, in the science department, it's different because we will discuss it thoroughly. Okay? We will unlock everything in that certain topic. And hopefully also I can um I can influence you with science to learn more about science. And it will explain everything. Okay? Again, it will explain everything. Even the phenomena, even the happenings in your body or the occurrence in your body, it will explain as long as you know the concepts. Right? Do you have any questions so far?
And none
None
so
so
far.
far.
I think that will be all for today, grade 10. Thank you for your participation. We did a lot
Thank you
of
as well.
translation. Have a nice day, grade 10. Goodbye.
Thank
Likewise.
you. Bye.
Bye-bye.
Bye, teacher. Have a nice day.